Good morning, Year 13. So, obviously this morning I can't be with you, which is a shame. Unfortunately, my daughter is a little bit ill, and so I'm having to stay home to look after her. But instead of doing the traditional textbook lesson, I thought instead I would record you one of these videos. Now, that hopefully will be useful to you, and more useful than going through the textbook, and hopefully by the end of it you should have a good understanding of the topic. Now, there are several limitations to the video lesson, obviously the first of which is that I'm not there to answer any questions, but from what I understand, unless the plans have changed, the teacher they've got to cover your lesson is a biology teacher, or at least somebody who has knowledge of biology, and so hopefully they'll be able to answer your questions. Now, if that's not the case, if they've had to change plans, then perhaps it would be worthwhile you writing down any questions you think of somewhere, and we'll go through them next lesson. Because it is important to check your understanding with me, or with a biologist. Second limitation is with the notes. I obviously talk quite fast. If you feel like you need it paused more often than I'm asking for it to be paused, then you should just stick your hand up and ask your teacher to pause it politely. I'm sure people won't mind. It's a thing. This video lesson is not ideal. And thirdly, unfortunately, it means you cannot do the required practicals today. I have to be there to assess any required practicals. So we're going to have to move that to 2 weeks from today. Please make sure you bring your lab coat to that. I will remind you closer to the time. But I apologise about that. Obviously that's outside of all of our control, but it is very annoying for you. Now, what you have then is you have effectively a revision lesson. We did last lesson on respiration. We're going to have an essay lesson, the second half of this double, which I know you'll all be excited about. I can hear the groans from here. But the first half of the double, I wanted to go through the light-dependent reactions of photosynthesis. Now, photosynthesis is actually a lot simpler at A level than respiration. There are only 2 reactions we need to know about, not 4. Only 2, and these are the light-dependent and the light-independent reactions of photosynthesis. Now, the light-dependent reactions of photosynthesis we think about happening first. They're all happening all at the same time. And that then produces the reactants for the light-independent reactions. And they are the bits that actually produce the glucose. So a bit like the link reaction, glycolysis, and the Krebs cycle, the whole point of the light-dependent reaction is to produce reactants for the light-independent reaction. I'm going to start off with these 4 questions as a starter. So I'd like your teacher to pause me here if you don't mind. Number 4, you will need to spend some time on, because it's a lot of detail there. So I'll give you a full 5 minutes. Please can you pause me for around 5 minutes or until everybody seems to be done. Thank you. Right. What are the products of glycolysis? Products of glycolysis are pyruvate. That's the classic product. That's the thing we need to know has to be produced at the end. ATP. There's a net production of ATP. So some people say ADP inorganic phosphate because they remember we have to break down ATP at the beginning. That's right, but overall we make ATP. So ATP is a product, and ADP and inorganic phosphate are actually reactants. And the final product is reduced NAD, which is one of the most important products. Where does the Krebs cycle happen? It's the matrix of the mitochondrion. You absolutely have to write the word matrix there. If you don't write the word matrix, you cannot have a mark, because at this stage we have to get into our minds that every different part of the mitochondrion has a different role. It's important to know what each bit does. What is the function of the thylakoid membrane? And do you know what? The first time I did this, and unfortunately I've had to record it twice because the first time the microphone stopped working, I gave you the answer to this question here. I'm actually going to save that answer. We'll go through that next slide because it can become relevant then. So hold that number 3 answer. And number 4, the function of reduced NAD. Firstly is to release its hydrogen ions and electrons. So release the hydrogen atom, which will dissociate into hydrogen ions and electrons with the help of various proteins and enzymes. The hydrogen ions then sit around while the electrons pass along the electron transport chain, which allows the hydrogen ions to be pumped from the matrix of the mitochondrion into the intermembrane space, and then to drain back by diffusion across ATP synthase to release the energy required for ATP production, to produce ATP. So you needed to write all of that: hydrogen ions and electrons released, electrons pass along chain, hydrogen ions pass through membrane and go through ATP synthase to understand the full function of reduced NAD. So, the title for today is, as you might imagine, the light-dependent reaction, because that's what we're learning about. Forget this starter for now. To be honest, we talked about it before. And that date is wrong because I'm recording this at 10:48 on Monday. It's actually, of course, Tuesday, 21st of September. Right. Oxidation and reduction. We talked about this in respiration a little bit, so I'm not going to dwell on this for too long. Now, you can really teach photosynthesis and respiration in either order. We tend to teach respiration first at Bournemouth School. It's not really relevant which. So we're going to just go over a few of the different bits, because you will find some parts of photosynthesis and respiration are almost exactly the same, and one of them is that they require reduction. Now, oxidation, we sometimes think oxidation is loss, but actually oxidation can be the addition of oxygen. That's why it's called oxidation. It's also going to be the loss of electrons or the loss of hydrogen. All of those things will be called oxidation. And overall that will give out energy. Reduction is where something loses oxygen, or we're going to talk about it in terms of gaining electrons and gaining hydrogen. And that takes in energy. So we're going to be ending up reducing several molecules, and that's going to be very important for photosynthesis. We need to remember what reduction is. It's the loss of oxygen. That's not really relevant for us right now. Or the gain of electrons and hydrogen. That is very relevant. Fine. So the first stage is to collect light energy. We have to get light energy for all of the light-dependent reactions. That's why they're called the light-dependent reactions. How do we absorb the light then? See, this is where I would point at somebody if I was in class. That's what you're going to miss out on. But hopefully everybody knows that we're going to absorb the light through absorbing it into chlorophyll. And that is GCSE. Chlorophyll is a molecule found in chloroplast that absorbs the light energy. And that's what chlorophyll looks like. Now there are actually other molecules which can absorb light, things like phytophytin or carotene, which are doing the same job. It's actually not just chlorophyll, which is why you can get red leaves and orange leaves, yellow leaves even. The only leaves you can't get are blue leaves, because blue light has the highest energy level in it. And so you have to absorb the blue. It's the main thing you have to absorb. So red, orange, yellow, green, fine, but blue, indigo and violet, you'll never find blue, indigo or violet leaves, because we need the blue light, because it has the highest energy level. And, interesting facts you can talk about over Christmas dinner. Right. Having got that we're going to need a photosynthetic compound of some kind, usually chlorophyll, now I want you to think about where will the chlorophyll be found. So I've got a chloroplast here, and we should recognise this as a chloroplast. If you don't recognise this, go away and revise your cell biology. You have to. And by go away, I mean stay in class, do this lesson, then go away. Right, so here's a chloroplast. We've got our outer membrane, coloured green here, that's nice. We've got an intermembrane space, yellow. We've got an inner membrane, which is blue for some reason. So it's a double membrane organelle. We know it's a double membrane organelle because we believe in the endosymbiont theory of chloroplast evolution, right? We believe that chloroplasts were originally independent bacteria that were absorbed by the original eukaryotic cells. So they have a double membrane because they had their own membrane, then they get absorbed by phagocytosis, and they have a phagosome wrapped around them. None of which is relevant for this lesson, but we should remember that from year 12. Inside this membrane we have the yellowy creamy stuff called stroma. Stroma is effectively the cytoplasm of the chloroplast. That's the way I think about it. And it's the same stuff really, though it does have a different biochemical makeup, to the matrix in the mitochondria. It's the same stuff in terms of function. Floating inside of the stroma here, just like in the matrix, are a load of ribosomes, because chloroplasts make their own proteins. DNA, because chloroplasts make their own proteins. And various other bits. The word plastic globule, not on the course, so forget it. But the main thing we have in here, the main structures, are these things called grana, which are these kind of circular things stacked on top of each other, and they're made of an extension of the inner membrane called the thylakoid membrane. They're joined together by these things called lamellae. This is one lamella, this long flat bit. And these are all big bits of the inner membrane called the thylakoid membrane. The whole thing is called a thylakoid. So if any of that is new to you, just do go and revise it. It's so important we understand the basics of cell biology from the beginning of year 12. It's a long time ago, I know, pre-second lockdown. Now, the question is, where would it make sense for the pigments to be? Have 30 seconds, talk amongst yourselves. Maybe the teacher could pause me until the discussion looks like it's getting boring. So where would it make sense for the photosynthetic pigments to be? Another advantage of video lessons, I can drink Fanta without feeling embarrassed. Right, so we've got then really two options people come up with this question in previous years anyway. The first is the outer membrane. That's the first idea. And that makes sense at first glance because it looks kind of green here and it's the outside, so that would make sense. The light would hit the outside, wouldn't it? That would make sense. If you think about it, if the light hits the outside and is absorbed, what is the point of all the inside? Because no light will ever get to it. So that's not what happens. And in actual fact, while it's coloured green here to allow you to see it, it's actually completely see-through, completely transparent. Is the outer membrane and the inner membrane too, both completely transparent. In fact, the chloroplasts are found on the thylakoid membrane, and that is because it has an enormous surface area to volume ratio, enabling it to absorb massive amounts of light for its volume, compared to the chloroplast's outer membrane, which, because it lacks quite a lot of surface area, it has a low surface area to volume ratio, really. Fine, so the photosynthetic pigments are found on the outside of the grana, on the outside of the thylakoid. So, we have this. So, this yellow bit here is equal to this yellow bit here. I don't know why it curves off, that annoys me, but that's fine. I suppose it's the curve at the end of the grana. Isn't that a nice thought? Right? So we're looking at the thylakoid lumen is in here, i.e. the inside of the grana, and the stroma is on the outside, i.e. the cytoplasm, if you will, background. So, embedded in the, excuse me, there's dead log length gone by, embedded in the membrane of the thylakoid membrane is a chlorophyll molecule here. Now, there are actually several different types of chlorophyll molecule. So if you're reading up about this outside the course, you'll come across chlorophyll A and B. We're just going to treat it as chlorophyll. It does do different jobs, I will grant you that, but we're going to think of it as chlorophyll. And electron carrier proteins, which you should recognise from the electron transport chain, and this molecule, which you should recognise from the electron transport chain, which is, I hear you all say ATP synthase. This is ATP synthase. So actually the layout is very similar to the electron transport chain. Now this is a good thing because a lot of the things I taught you in the electron transport chain can be directly copied and pasted to the light-dependent reactions. It's a bad thing because in tests people mix them up. Please don't mix these up. They're different. They do different functions. They do a lot of the same things, but they have a different function. So, step 1. And incidentally, I'll give you time to write these notes after I've talked through them, so just follow me for now. Don't try and write notes while I'm talking. Step 1. Step 1, there we go. Light. Okay, light hits chlorophyll. Of course, light hits everywhere. It's just chlorophyll that absorbs it. Chlorophyll absorbs a photon of light. Do I care that it's a photon? Not really. It is a photon. No one cares. Because in actual fact, millions of photons will hit at the same time, and so whether it's 1, 2, or a million is kind of irrelevant, particularly at A level. What's going to happen here? Well, actually two things are going to happen. And we'll do the first one now and the second one later on. The first one is something called photoionization. Photoionization. Might even, when you write question point 1 here later on, be worth writing the word photoionization next to it. Because this is where we use light, photo, to remove electrons, thus turning chlorophyll into an ion. Because electrons are negative. Electrons are excited. For what it's worth, it's 2 electrons. Is it relevant? Again, no. Forget the numbers. Electrons are excited. What do you reckon is going to happen to those electrons? Well, hopefully everyone is thinking of. The electrons are going to be passed along the electron carrier proteins, just like in the electron transport chain. Like that. Okay. So, If your teacher could pause me there, that's the first 3 out of 8 steps. So I will give you 4 minutes maybe, but let your teacher see how long it takes you to write down those facts. And remember, write photoionization next to step 1. He points as if you can see it. Right. So, that's that. Now there's questions which I want you to have a look at, but not do. So, question 1, what happens when light energy is absorbed by chlorophyll? Have a think about it. What happens when light energy is absorbed by chlorophyll? To be fair, because I'm not there, I want to have a look at all the questions at once. Have 30 seconds. Maybe your teacher will pause you, and I'll give you the answers. Okay. Right. What happens when light energy is absorbed by chlorophyll? Well, it excites 2 electrons, or just electrons, called photoionization. Where do they go when they're excited? There's a joke in that probably, but they go to the electron carrier protein. Okay. What happens as they're passed along a chain? The energy level drops, thus releasing energy. And I hope everyone can predict what's going to be used, what that energy is going to be used for. Well, there are hydrogen ions just hanging around. There are always hydrogen ions hanging around in the chloroplast. They just exist. We're going to talk about where they come from later on. These hydrogen ions are needing to be pumped across the membrane to power ATP synthase. So, quote step 4 here, each time the electrons move down an energy level, they release a small amount of energy, and as if by magic, this will happen. Now, I believe that's that, yes, so have some time to write step 4 there out of 8. Again, apologies for all the pausing, thank you to the teacher who's doing this. Right, so each of the electrons move the down energy level, they release a small amount of energy, which allows a hydrogen ion to pass across the thylakoid membrane. Now, I would spend more time on this if I hadn't taught you exactly the same thing on the electron transport chain. So have a look at the questions there. There's probably not even a need to pause me because I can just talk you through these as we go. They're exactly the same. What gives the energy for hydrogen ion to transport across the thylakoid membrane? That is the electrons being passed along the electron transport chain. Where in the chloroplast is the highest concentration of hydrogen ions? That would be the stroma of the chloroplast. Why did I say that? I apologise. Of course, it's not the stroma of the chloroplast, it's the thylakoid lumen. We're passing them out of the stroma into the thylakoid lumen. And so always, because we're continually pumping them across, there's always the highest concentration in the lumen. Fine. Now, this is where I stole this slide from the respiration topic. And this is about ATP synthase. Now, again, I would dwell longer on this if we didn't already know about ATP synthase. But to remind you, as hydrogen ions pass through this, they physically turn this kind of turnstile thing up here. That gives the mechanical energy, which forces ADP and inorganic phosphate together, reducing the energy required to form ATP to the point where it's an energy positive process, and we release ATP, which has more energy than what the ADP and the inorganic phosphate had. And it looks a bit like this. I always love this. Is it a GIF or a GIF? I don't know. Either way, I could watch it all day. And if it is physically turning, that is what is happening, thus allowing the ADP and inorganic phosphate to release ATP. If you don't get any of this, look back at your ATP synthase notes for respiration. I'm not going to get you to write this down because I'm fairly confident I got you to write down the exact same thing for respiration. Right, so next step, exactly what you'd think it would be. Hydrogen ion goes through by diffusion because there's a higher concentration of hydrogen ions in the thylakoid lumen than there are in the chloroplast stroma. So through they go, and again, and bam, ATP. Okay, so I'm very proud of that. I animated that myself. Now, this produces ATP. Now, you may immediately say to me, hang on a minute, respiration makes ATP. I know photosynthesis doesn't make ATP, and you'd be right. This ATP is going to be used later on, so overall we're not going to make ATP. This is going to be made briefly now, and then used later on in the light-independent reactions. But for now we need to make some ATP. So that's great, we made ATP. Fantastic. Good. I have an issue, which is that the electrons are clogging up the electron transport chain. Now, if this was respiration, what would happen to them? Where would they be attached to? What is the terminal electron acceptor in respiration? I hear you all shout oxygen. Oxygen is the terminal electron acceptor in respiration. In this case, there's no oxygen around, or not yet. So these electrons have to go somewhere else. Where else could they go here? They could go to these hydrogen ions, couldn't they? And this is what happens. And instead, I will give you time to write this note in a second. Electrons go to hydrogen ions and make hydrogen atoms. Okay. These hydrogen atoms, they don't just hang around. Hydrogen is not a product of photosynthesis. In fact, they're needed again in the light-independent reactions, and again, they're not allowed just to float over there because they would just diffuse out of the plant. They're attached onto a molecule called NADP to form reduced NADP. Fine. Now, NADP and NAD, classic mistake people make is mixing them up. Now, the way I remember this is P stands for photosynthesis. It doesn't really, but that's the way I think about it, because if you remember P stands for photosynthesis, you can never write NADP in respiration. People do, though. Please don't. NADP is only found in photosynthesis. NAD is only found in respiration. Fine. I understand why you get them mixed up, but the P is for photosynthesis. If you just remember that, that's fine. As long as you know it's not actually true. Right, question, so point 5, you need time to write that down. I don't think I gave you it earlier, so again, if your teacher wouldn't mind pausing me. Right, point 6 and point 7, I think can be put on the board together. No, they can't, annoyingly. So write point 6 quickly there. Again, unfortunately, I'll have to be paused. This is where the video lesson thing falls down, unfortunately. And point 7 here, the hydrogen atoms we needed later on. So they are attached to a hydrogen carrier called NADP, forming reduced NADP. Again, for final pause here, would you mind pausing me, and we will just write this note. Fine. Now this is perfect, because I need ATP for the light-independent reactions, as I will find out later, and I need reduced NADP for the light-independent reactions, as I will find out later. So far so great. But there's an issue here. But before we come to that issue, here's 4 more questions to have a look at. Again, unfortunately, pause me and give the students some time to discuss these, if you wouldn't mind. Just a couple of minutes. Right, so points 6, 7, 8, and 9 are, what's the name of the protein channel which is used to create ATP? This should be ridiculously easy for you. If it isn't, you have to go back and learn respiration. Because we're not going to touch on respiration much again, and this is a respiration question effectively. It's ATP synthase is the answer. What's passed through this channel in order to produce ATP? Hydrogen ions. Hydrogen ions are. What happens to the electrons that are still in the electron transport chain? They attach onto the hydrogen ions left behind to produce hydrogen atoms. And what's the name of the carrier molecule which takes them away? NADP, or reduced NADP once it's been reduced. If any of the first 2 questions is not familiar to you, please come to clinic next Monday and I will help you with respiration. But please don't just sit on this because you will struggle on the next test, unfortunately. Fine. Right, there's an issue here. Have a look at that and try and tell me what the issue is. Think back to the beginning of the slide, of the, whole thing. Tell me what's the issue with this whole thing. What do you think? Hopefully no one's putting their hands up because I can't see. Well, we've got to think, ATP. Great, I've made that. That's great. I wanted to make it, I've made it. Fine. Reduced NADP, whoa, excuse me. Gone backwards. Sorry, gents, ladies. There we go. The reduced NADP, need it for later. Great, made it. But if you remember, way back at the beginning, we actually started with 4 hydrogen ions, and now we've got 2. Now losing 2 hydrogen ions makes no difference to a plant cell whatsoever, but you can't want to keep doing that forever. Eventually you'll run out of hydrogen ions. And perhaps an even bigger issue, I lost 2 electrons from the chlorophyll, and I haven't replaced them. 2 electrons, is it going to make a huge difference to a large molecule like a chlorophyll? Well, possibly, actually, but maybe not. We can't keep losing 2 electrons over and over and over again, because you'll run out of electrons. And chlorophyll will become really positive and start attracting ions all over the place. It'd be ridiculous. Coated in chlorine, I don't know, whatever. Anyway, regardless, you have to replace those 2 bits, the hydrogen ions and the electrons. Now, if you think about the word equation for photosynthesis, carbon dioxide plus water makes glucose plus oxygen. Could you spot anywhere a reactant that might have hydrogen ions and electrons in it? Hopefully everyone screams water at the TV, right? So water is what we're going to use here. It's the first reactant we're going to use up. So along comes some water. It's minding its own business, it's not noticing what's about to happen to it, and bam! Violent process this. Now this is called photolysis or photolysis because it's photo for light and lysis, like lysosome, like lysosome, which is breaking down things with light. We're breaking down water with light. Photolysis. Break it down into hydrogen atoms and electrons. Now the electrons, they are absorbed. They're absorbed by the chlorophyll. The hydrogen atoms just hang around and rebalance everything. And this oxygen, well, that's great because if we did this twice, we'd have 2 oxygen atoms, make an oxygen molecule, and that will be one of the products of photosynthesis, oxygen, which is fantastic. Okay, so we've used up a reactant, water. We've made a product, which is oxygen, so we must be getting somewhere now. So again, unfortunately, you will need a few minutes just to write these notes. So do pause me, please. Right. Okay. So that's it. That's the light-dependent reaction all done. Now, you'll remember I said there are there are 2 major things you'll be asked about any of these chemical reactions, and 1 minor thing that you might be asked about. The 2 major things are, what are the reactants and products? You usually get asked one or the other. What are the reactants and products? And where is it happening? Those are the 2 things you'll be asked a lot. Where does it happen? What are the reactants and products? The final thing you can be asked about is the whole process which we've been through. That's why we go through it. Now when it comes up, it's usually worth something like 6 marks, which is fantastic. But mostly you'll be asked 1 markers. What's the reactant here? Fine. So, what are the reactants of the light-dependent reaction? Sit and have a think about that. And what are the products? And if you wouldn't mind writing that down, reactants, blah blah blah, products, blah blah blah. Light-dependent reactions. I don't know why I'm sitting silently, to be fair. Hopefully you've been paused and played by now. So reactants. Give somebody time to pause me if they haven't already. Reactants are, firstly, ADP and an organic phosphate. Secondly, NADP. And thirdly, water. In that order in terms of our diagrams. Light isn't a reactant because reactants are chemicals. Light is not a chemical. The products, ATP, reduced NADP, and oxygen. Okay, so that's the end of that. Right, now I'm going to finish off with an exam question on this topic. It's going to be quite a nice long answer one. I have to pull it up from Exam Pro, so I'm going to pause myself, and I'll grab it and put it on the board for you. So this is a beautiful question. It's a real beauty. Now, part B here is what we're looking at. Forget part C down there, dry mass of plants, blah, blah, blah. This is a really nice question. If you get a question like this, you can feel really pleased. It's the kind of thing you will see towards the end of the paper. Long answer questions. Now remember, paper 1 always has 10 marks of long answer questions at the end. Paper 2 actually doesn't have that necessarily, but it will have long answer questions. They can just be scattered throughout. So have a look at that. 6 minutes for 6 marks. You may not need that long, excuse me, because you can bullet point this because you've already written notes about it. I think for the final time, thank you to the supply teacher. Would you mind pausing me? Right, 6 marks here then. So, the role of the electron transport chains. Forget part A there, that's actually about the light independent reaction. Electron transport chain accepts excited electrons. It's obvious, you have to write it. From chlorophyll, or photosystem. Forget it. We'll come to photosystems another time, not right now. Electrons lose energy along chain. Great. ATP produced. Whoa, we've skipped some stages. Okay, the stages can vaguely come along down here. They've kind of rearranged some things. But ATP produced, a mark. From ADP and anorganic phosphate, a mark. Reduced NADP formed when electrons and hydrogen ions combine with NADP. Hydrogen ions from photolysis. Now listen. Some people will only have got 3, maybe 4 marks, probably the first 4 out of 6. With a 6 marker, you have to write down everything that is related to the question. It's like that reduced NAD question I asked you at the beginning. You can't just write, well, reduced NAD releases hydrogen ions and electrons. Why does it do that? What happens because of that? Where do they come from? Write everything. With a 6 marker, write everything. Right? So you've got 8 marks there. I would have expected you to get all of them, because they're all quite obvious, but it's about making sure you write down everything that could possibly come up. Right. Hopefully that's nice and straightforward. And I will end that there. Now we're going to finish off with an essay question. The essay question, I'll find out what... Right, here's the thing I use to plan my lessons. So the essay we were going to write is an essay on the importance of enzymes in plants and animals. So once you've got this light-dependent reaction, you can now start this essay. Now, I am not sure how long you'll have left of this lesson. It might not be very long at all. Should have about 45 minutes for a complete essay. So, depending on how long you have left of the lesson, or depending on how many paragraphs I want you to write here. If you've only got 25 minutes left, or 20 minutes, I want you to write a 2-paragraph essay. If you've got 30 to 35 minutes left, I want you to write a 3-paragraph essay. If you've got 40 minutes or above left, I want you to write a 4 or 5-paragraph essay. So, have a look at how many minutes you've got left. That's the length of essay I want you to write. If you have less than that, draw me a plan for this essay. Hopefully you have more time than that. I don't think I've been paused that many times. It's a really nice, easy one, except one thing, I'm going to order you to include either respiration or photosynthesis. You all know at least one enzyme involved in both of those things. So write down a nice paragraph about that if you wouldn't mind for me. And we will mark them together next lesson. Okay? So the rest of the lesson should refer to that. Now, that's the end of me. Usually I just sit at the front and pretend to work while you write your essays. In actual fact, I'm going to go to bed. So thank you to you for putting up with this video lesson. I know it's not the easiest thing in the world to follow. And thank you to the supply teacher who has paused me so many times, no doubt magnificently. And I will look forward to seeing you all whenever our next lesson is, when we will either do the light independent reaction, or if we get time, we'll do Required Practical 9, as was planned. I hope you have a great morning and a great rest of your day, and I will see you soon. Goodbye. That's this button.